Hey guys, welcome back. So today we're starting our last chapter of the semester, and this will be kind of adding everything together that we've learned about in chapter 17. We'll be going through not just gene expression in terms of transcription translation, but adding on to those to allow us to really fine-tune gene expression so that we can get the exact level of gene product function that we're looking for. So many people think that gene expression is either on or off, but in reality, this is more of a spectrum. So you can think of it more like a dimmer switch that can range from high expression to low expression. So anywhere in between, allowing the cell to control exactly what level of gene product that it's looking for. So remember from our last chapter, we talked about the idea of gene expression, meaning not just the sequence of DNA, but exactly what type of mRNA or protein we're looking for. So it doesn't matter so much about the sequence of DNA, we're looking for the production of some gene product. So gene expression can really be controlled at many different levels in the cell. So most of these we've talked about before, but we're just going to add some new elements today. So we can control whether a gene product is produced at the DNA level. So back to chromatin modification that we learned about in some of the first chapters. We can definitely see gene expression being controlled at the transcription level. So this is one of the major places where people talk about gene expression being controlled. This is basically, did I actually transcribe this gene? Did I make this RNA or not? We can also add RNA processing, specifically RNA stability related to alternative splicing, sometimes the poly A tail, and there are a couple of other things that we can talk about with RNA stability. Next, once we've exported this into the cytosol, we can actually use that RNA stability and some of these properties to even control whether we translate it or not. So that's right, you can go through all of these steps for transcription and then not necessarily even translate this mRNA right away or at high levels. It can also allow you to get rid of this fairly quickly. Once we have a protein, there are a few different ways that we can control it. Okay, so protein processing. Alternatively, we can regulate whether we're actually going to have a protein at all. We can get rid of it if we need to really quickly. And we can also modify its function with inhibitors or activators, other feedback loops, and other modifications, which we'll talk about at the end. So we're going to go through each of these and talk about some examples of ways that these can be controlled. So let's start at the DNA level. Chromatin modification. So chromatin modification is going to be related to, again, that sort of packing structure that we talked about with chromatin in chapter 6 when we talked about the nucleus. So if you can't quite remember these structures, be sure to go back and look at chapter 6 in the video and slide set where we talk about the nucleus. And so today we're going to build on that, thinking about how we can control the compactness level, how compact this is going to be and how loose it's going to be, so that we can give access to our transcriptional machinery. So it's all about within kind of a range, kind of tightening and loosening this chromatin, depending on whether you actually are going to want to transcribe this or not. So there are a couple of different modifications that we're going to talk about. In reality, there are many more than this, and it's actually quite a complicated concept in terms of how many different types of modifications can sort of stack on top of each other to form signals. So we're just going to focus on the more kind of simple, generalized ones today. And we're going to talk about these histones. Histone acetylation, so adding acetyl groups to our histone tails. Histone methylation, adding a little methyl group to these tails. And we're also going to talk about doing some methylation on this DNA, the sequence itself. So let's just kind of remember about histones. Our histones are going to be kind of in groups. This is an octamer of our histones so that we're able to form our nucleosome with the DNA. And they do have these little tails. So these are proteins that have tails, and the DNA loops around it. When we talk about modifying histones with acetylation or methylation, we're going to be adding these groups to the tails. Okay, and again, what we're aiming for is to control how tight or how loose this DNA can be. So we're looking at a very detailed structure here. There's not really a lot of things you need to memorize, but I think this gives a really good idea of what's going on here. And you can spend some time looking at this. Here's acetylation marked with a little A. We've got different types of methylations happening here, different amounts of methylation. And you can see that there's a lot of acetylation up here, that kind of thing. And this is where we're going to be adding those modifications. Again, you don't have to memorize this. We're just talking really generally about this today. Just think this is a really kind of more informative view of what's going on. So we can add various modifications to these proteins, and we can either mark them as active. So active chromatin is going to be more loose, and we're going to refer to this as euchromatin. Okay, so euchromatin. And then other times when we don't want gene expression to occur in a region, we can tighten this down, sort of lock it down, and use some of these modifications to mark this area as repressed. We call these sort of tightened up compact regions heterochromatin. Okay, so we can think about, again, adding acetyl groups, methyl groups on these tails. And generally speaking, acetylation is going to mean more active region. So again, everything in the sort of top part are active markers. Notice there's just quite a bit more acetylation going on up here. And again, generally speaking, we kind of attribute this more heterochromatin state to methyl groups. Okay, you can see it's not 100%. There is methylation going on down in this repressive marker, but there are other things too that we're not going to talk about today, like smolylation, etc. There's some methylation going on up here too. Okay, in reality, it depends on exactly where these methyl groups are and what they appear sort of next to, what their neighbors are going to be. This forms sort of a histone code. So this is kind of like reading words biochemically. Like you can ask somebody whether, the letter N is going to be a negative or a positive thing. Well, it depends on what letters are next to it. If it's like N next to an O, then no, that's the word no. It's not going to be, something necessarily that's affirmative. So it just depends on context, what's going to be next to it. But for our purposes today, we're going to think of methylation as being more repressive in nature. By contrast, acetylation will be more activating in nature. And we can see why that actually is. When we have a lot of acetyl groups on these histone tails, you can see that this is going to kind of loosen up. We've got a lot of access. If you remember back to chapter 17, transcription is a lot more complicated than we ever thought. So we're actually worried about getting all of those general transcription factors, the TF2D, the TF2AB, all of those guys have to fit here with the RNA polymerase. So allowing it to have kind of a large access space, okay? And then if the DNA is tightly packed, we do not allow access to that. So this is why we think about methyl groups kind of tightening this up. This is why our tightened chromatin makes it more difficult to express and transcribe these genes. Okay, and you can see I did pick a nice Adobe stock photo, but that's fine. So we've got acetylation contributing to looser, more accessible DNA, methylation going to be attributed to more inaccessible. Okay, so we talked about two things we can do to histones, acetylation, methylation, but we can also methylate DNA. Okay, so what does that mean? Previously, those modifications were happening only on histone tails. Okay, so they were not happening, anywhere else. They were on the protein. When we talk about DNA methylation, it's literally what we're saying it is. It's going to be a methyl group that actually occurs on the nitrogenous base, largely of cytosines. Okay, so this is the cytosine nitrogenous base. We actually add a methyl group to it. Okay, and when we methylate the DNA, it has the same general effect that we saw with methylating histones. So tightening this down. Okay, and one kind of difference, again, a lot of these are very general because we're not spending a lot of time getting into the details, but one kind of, general difference between our methylated DNA and our methylated histones is that histone modifications can be extremely transient. We can be sort of like moving these around, repositioning acetyl groups, methyl groups all the time. In fact, a lot of acetylation and methylation happens according to circadian rhythms. So when we get ready to express a gene at nighttime, we can go ahead and open that chromatin up and then close it back down in the morning. So it's just really happening all the time. Methylated DNA tends to be found in regions that we're more interested in shutting down long term. So this can be found in things like development. We start to think about cell fate when a cell decides, hey, I'm going to become a muscle cell instead of an eye cell or any other skin cell, any other type of cell. So we don't need eye cell genes. We don't need skin cell genes because we're going to become a muscle. So we start to tighten those down kind of permanently. So I tend to think of methylated DNA as being associated in general with regions where we're going to be a little bit more permanent when we shut things down. Okay, and you can of course combine DNA methylation with any of the other histone modifications too, so that's always fair game. Okay, so we've talked about at the DNA level how we can adjust transcription and translation, okay, or just transcription for now. Sorry, gene expression leading up to translation of our gene products. Okay, so we can control transcription here, but we've also got other things that we can do. So the first step is if the DNA is closed down, we are probably not even going to bother with that. But if it's open, then we have to worry about some of these next decisions. How much are we going to make of this RNA? So we've talked about a couple of other things going on. Regulation of transcription initiation is going to be a major point of control. Okay, so how do we actually initiate this? We talked again in chapter 17 about assembling all of those nice general transcription factors on the promoter, the TATA boxes in here for eukaryotic cells. We can have what are called proximal control elements. They're called this because look how close they are to the promoter itself. Okay, you can also have distal elements or enhancers that are going to be way upstream sometimes of our core promoter. So this can be 10,000 nucleotides away from where we're actually going to care about this. Then of course we have the rest of the gene structure, the protein coding region, termination region, so that we are ready to actually make this protein. Okay, so of course, again, we talked about general transcription factors. Again, that's that like TF2, D, A, B, F, H, E, those guys, all of those are general transcription factors. So if we open up the chromatin in step 1, then we're allowing those general transcription factors to come in. But to really crank up expression, we're going to have to do a little bit more. Okay, and that's going to require, again, the help of our proximal and distal control elements. Okay, so again, we learned about the promoter before. Here's our TATA box, and this is going to be where RNA polymerase will actually start transcribing. Okay, this is right next to the gene. But we can have upstream these distal control elements, enhancers. Okay, and it's important to note that the enhancer, these distal control elements, are part of the DNA sequence. So they're not protein coding. They're not going to ever be contributing to what we tell it to do as in terms of making a protein, but it is going to be a sequence that's specific, that's going to help us activate things. Okay, and specifically these can be recognized by proteins called activators so that we're able to ultimately activate transcription. So the role of all of these, the enhancer DNA sequences plus the protein activators, is actually to facilitate the assembly of the pre-initiation complex, that sort of general transcription factor complex with RNA polymerase II. This is going to be facilitated by our enhancer and our activators. So that's the whole role, is to help crank up expression of these beyond basal or just baseline levels. So why does this work? Why does this even happen? Why do we need this? Okay, so there's a lot going on here, seemingly, but it's okay. All we're going to do is mess around with the shape of this DNA to help facilitate, again, those general transcription factors binding. So notice we've had our activators bind to the enhancer. We've got a DNA binding protein in here. This will make a little bit more sense in just a second, okay? So the activators themselves are proteins that recognize the DNA sequence of the enhancer, and they go ahead and attach there. So they have to have some type of DNA binding domain that recognizes a specific sequence in order to bind there. So this can result, when we get down to this binding portion, in even more accessory proteins. So our DNA binding protein, mediator proteins. So this is a whole group of them, all right? And so what we're going to see in a second here is that these mediator proteins are going to facilitate sort of binding between our activator proteins and our core promoter, okay? And so then the general transcription factors can come in and assemble. So let's see all of these kind of come into place, okay? So we can see that our mediator proteins and activators, notice these general transcription factors are kind of aided by the fact that these activators are here and help them sort of situate on this core promoter, okay? And that allows, of course, RNA polymerase II to come in. So once RNA polymerase II is recruited in, we can go ahead and begin transcription. So just worth noting, this seems, again, like, okay, these mediator proteins, I can see why these might be helpful. DNA binding is a really common way to increase the amount of transcription that happens. So this happens for quite a few different genes, and cells have different mechanisms for kind of ensuring that this becomes bound. So in this sort of DNA bint conformation. So again, this is just something really common that we see. And without these enhancers, again, we might assemble the general transcription factors here, but it might not even be at meaningful levels that are biologically relevant for the cell. So we may produce several molecules of RNA, but it's not enough to really get any job done in the cell. So if we really want a lot of RNA, we're going to have to do something extra to crank this up. That's where the enhancers come in. Okay, so is there RNA? So we can think, step one, we had to loosen the chromatin to allow access for all of this stuff. You can see with all the stuff here, you're going to need some space, okay? And then we have to facilitate or enhance the binding of these general transcription factors. That's step two, to get RNA polymerase to come, okay? And of course, we can control levels of transcription based off of what's going on with these enhancers. So that's kind of step two. Did we even make our RNA? So let's look at a biologically relevant example, thinking about different types of cells. So the example that we're going to use is going to be the liver cell and the lens cells of your eye. So notice what's going on here. We have promoter for a certain type of gene called the albumin gene. The albumin is going to only be produced in liver cells. So this is liver cell specific. Whereas crystalline is going to be produced in the lens cell of your eye. So all of our cells have the same DNA. Your liver has the same DNA as your eye. But how do we get a situation where only the liver is producing albumin and only the lens cells of your eyes are producing crystalline? Okay, so that's where some of these control elements, these enhancers, are also going to come into play. So we can look at a couple of things here. So notice that the enhancers have some similarities They both have a gray section, okay? But our albumin has kind of a yellow and red, whereas our crystalline has an orange and pink. And these color differences represent different sequence differences. So this gray sequence is the same, but the yellow and red sequences, the orange and pink, are different. That means that they will recognize different proteins. So this is going to allow different sets of proteins to bind to them. Okay, so we've almost got the setup. Let's see this start to go in action. Okay, so in addition to the different enhancer sequences, each cell type is going to have a different set of those activator proteins present. Okay, and so hopefully you're probably going to guess that they'll have the appropriate